Hey, hi everyone. Welcome to this semester. How are you guys doing? How was your weekend? And how are things going? So, welcome to Introduction to Microbiology. So, this is for spring 2026. And in this course, we will explore the hidden world of microscopic organisms and their impact on our lives. So, microbiology isn't just about pathogens, it's active research. Here we will see examples like Streptococcus gordiani and how it, it forms biofilms basically by adhering to saliva-coated surfaces. And also, we could also see examples from Acinetobacter baumannii, which also requires iron and zinc to survive in their host. We also see factors that impact pathogenesis of a pathogen called Francisella, and basically factors that affect its survival and its activities. So throughout this course, I will be available. Currently, I've not been able to set out the model page, but I plan to do so. There's just a bit of delay, but I'll be doing that throughout this week. With that being said, I'm not always on campus. For that reason, if you need to access me, you should email me through my email, which is posted right here. And also, office hours are by appointment via email. And then you can send me email throughout the whole day. I'll check it out and reply you. And if you want like a one-on-one Zoom meeting around mostly after 6 p.m. works the best. If not, then we have to do it on weekends or during in-person after class. So, since this is asynchronous class, mostly I will have asked you guys to introduce yourselves and then talk to each other. But basically, since this is an asynchronous class, I will just say that and you guys can do this when we come back in person in lab, and then we can basically do this. So this course, we will have two different pages, which is the model page, page orientation, which you have access to both the lecture and the on the lab. And then we have Connect, where you have e-textbook and also the smart book to work with. There are assignments which basically is pre-class assignment. You have to read and work with the smart book assignment. This is mostly done individual and you have to complete it as we cover new materials in class, and it's due by the associated exam. And we have the post-class assignment, and this is actually also done individual. It applies to microbiology case studies and some which are fit integrated. And then we have the extra credit assignment, which you can do it individually or with a partner, and basically you have to review and then apply materials. And then we have unit exams and or basically the final exam. So lab will start on Tuesday. And during Tuesday, please make sure you bring in your lab one handout, which should be on Moodle. And then we also be working on lab. During the lab, you also have to make sure you bring in your lab coat. You have to adhere to all lab rules, which is not bringing in food, wear clothes, that kind of wear closed shoe toes, toe shoes, and make sure your feet is covered. Make sure you wear clothes that cover yourself. Definitely bring in a lab coat, which is a requirement. For gloves, we will provide for you. And our lab is on Tuesday, 7:30 a.m. And then we also have the evening lab, which is at 6 p.m. if I'm not mistaken. And then there are like 10 banks and practice questions and other resources which are available. And in this class, we have an AI agreement, which basically means AI can help you learn, but you shouldn't rely on it and paraphrase or let it do all your you shouldn't rely on it and let it do all your assignments for you, which is actually the cross line and it's illegal and will never be tolerated in this class. So basically, food for thought. So In this course, we consider how science and faith intersect. And here I want you guys to consider the words of Francis Bacon. He says, "A little science struggle a man from God, and a lot of science bring him back." Simultaneously, I also want you guys to pay attention to words from Louis Pasteur, which is basically what he said was that he felt like that studying nature increases his amazement at the work of the Creator. This is basically food for thought. So in this course, or basically today, the objectives that we are going to learn today is to list the major type of microorganism, included in the study of microbiology. And we also want to learn how to correctly write the binomial name of an organism. And then we also want to match scientists with the microbial discoveries that each are accredited for. So microbiology is just an area of biology that deals microorganisms too small to be seen with the naked eyes. So we have microorganisms or microbes. These are basically organisms you can see, and they will include things like germs, viruses, and agents in bags. And one example of this can also include parasite. And one of example of things that we can't see in our eyes is basically this parasite, which is the Lualua parasite, basically an African eye worm by most people, and it's caused by I'm sorry, let me take it again. This worm which we see, oh, I think you guys cannot see because I don't have a pointer. Okay, now a pointer is showing. Basically, this worm, Lualua, causes a disease called luasis, basically called an African eye worm by most people, and basically it causes the disease Lualua and is passed on to humans through repeated bites of deer flies. And so basically from here we can appreciate how microbes can never be seen by the naked eye, but we have to see them through the microscope. So to move forward, we also have to understand that microbes have to be grouped or are grouped into major groups. And then one of these major groups, or the major groups that we have to understand, is eukaryote. And then under eukaryote, we have hermits. Basically, these are multicellular animals like worm-like pathogens, both harmful and then harmless. And some of them we can see from this image like a worm, basically looking at someone's gut. And then here we can see this parasitic worm outside of the human body. Basically huge, you can clearly see it. And then we have some which we classify them or under the group called fungi or fungi, depending on who is pronouncing it. And these are macro or sometimes microscopic forms, and they consist of yeast, and they can also be both harmless and pathogenic, depending on where you find them. And these are some images. This is being seen on a corn or a maize, as you can see from here. And they are usually like spore-like or like more like a foamy-like in nature to see. And then number 3, we have the protist, basically made up of protozoa and algae or algae, also based on who is pronouncing it. Mostly single-cell organism, both harmless and pathogenic. And here in these images, we can actually see an example of them. And then on the next page, what we can see is also the next, which is prokaryote, which consists of bacteria and archaea. And then they are also single-celled, harmless, and pathogenic, depending on where you find them. And this is microscopic images of them. And here, the point 5, or basically down here, we have viruses. So viruses are basically non-living particles of proteins or nucleic acid, and they are mostly pathogenic. I haven't come across a virus which is not pathogenic before. Probably you might, but I don't know. But basically, I don't think it even exists. They are mostly pathogenic. And then the last under this group of prokaryotes is prions. These are non-living infectious proteins. And here, I want us to do just a quick brain test. think of it very carefully. Which of these groups are not true microorganisms? Which of these groups under here, bacteria, archaea, viruses, prions, which of these groups are not actually microorganisms? The answer is viruses and prions. And then in the next slide, we'll talk about why they are not actual microorganisms. So since we have different groups or subset of microorganisms, then we needed, or scientists needed, a way to classify them or to identify them and talk about them. So early microbiologists classified them by shape, arrangement, growth, characteristics, and habitat. And there was also a disadvantage or a setback in this method because many microbes were unculturable. So if you can't culture them, you can see them, you can see how you can study their characteristics. Until recently, that we have newer methods of characterization, which encompasses of that of biochemistry, molecular biology traits, and then genetic ribosomal RNA or genetic sequencing, which is mostly being done by the thermal cycler of which we use for qPCR, basically amplification of genetic material. And then this has actually helped us in the classification of microbes. However, though viruses, which are not like organisms, also have to be classified, but they also have their own classification. So to fully understand classifications, we all follow a hierarchy from domain. Historically, all prokaryotes were lumped into kingdom Monera, though modern classification is quite complex. So as you can see from here, that we have the kingdom Monera, and under it we have domain bacteria, domain archaea, domain eukaryotes, and we've already discussed everything all under it. But we also have to understand, since we said viruses are non-living and we can actually classify them, then the question is, where exactly are we going to classify viruses into, or any of these three? And the point is, because they are non-living or they are considered non-living, they cannot be classified under any of these three domains. For that reason, we often use the Baltimore classification system, which categorize them based on the type of nucleic acid they are expressing, whether RNA, DNA, and their mechanism of replication. So this you can actually have to study. You actually have to use the thermocycler or the PCR to actually understand this. And for that reason, viruses are studied within microbiology as infectious agents, but occupy their own unique non-kingdom classified category. So now the question is, we already know, now we have to classify, we'll be talking of classify. How do you classify an organism? And to fully do this, we have certain rules that we have to obey, which these rules fall under, these rules were acquired when we had the binomial nomenclature, basically a way of assignment of a scientific name to an individual organism. And you have to follow these rules in order to be able to do that. So under these rules, we have levels of classification, which is domain, we have kingdom, and then we have division, followed by class, order, family, genus, and species. So every single living organism is actually classified scientifically under all of these under all of these five categories. Under all of these categories, and they all fall or they all followed the binomial nomenclature way of naming an organism. And there is a certain rule which guide this binomial nomenclature. Basically, they always have to make sure that the genus name is capitalized from the beginning, and then the species name is always written in smaller letters. So as we can see from here, this organism, which is the dog, is Canis lupus, right? So we can see from here, Canis lupus, The C is capitalized, basically the genus name, and the species name is lupus. And then there is also another rule, which basically means you can italize them in a print or underline them by hand. Or you always have to make sure that since you have already, this is already known, the Canis lupus, you can always abbreviate the genus name and then you write the species name, which is also widely acceptable. So which or what rules for binomial nomenclature do you think is actually useful? And you have to think in pair and you can share it. We are not in class, so you can find someone to discuss with. So this is actually a test to see if you understood what I explained earlier. So I'm go over the rules again. Make sure genus name is capitalized, species name is small, and then you can also write the genus name. You can abbreviate it, and then you write the species name. So back to this page again. Which of these names do you think is correct? So you can take some time, think through, and then give me an answer. The answer is D and G. So in D, you can see we have the genus name here and the species name. Genus capitalized, species name well written. And in G, you can see here, you write a simple P to represent the Pseudomonas, and then this is the species name. There is more to practice on Connect if you read the course material. So microbes often have variants that we call subspecies, strains, or stereotypes. So for example, the microbe Escherichia coli, which is basically a harmless gut bacteria in most humans, we all have them in our gut. Their subspecies or strain or stereotype, basically, we have three, which is E. coli K12, which is common lab strain. We'll actually be using some of them probably in this semester. And then we have the UPEC, which is basically a strain that causes urinary tract infections, UTIs, and I hope none of you get this bacteria because basically you've contracted a UTI. And then we have the E. coli 0157H7, which is basically a stereotype that causes food poisoning. So hopefully none of you get the other two, these two stereotype of E. coli, but this one is harmless and we all get to use it. So basically, a stereotype represents a species that stimulates a distinct pattern of antibody response because of distinct surface molecules. So they are under the same species, but they are different based on the distinct molecules they present on their surfaces. So does anyone know of any bacteria which is beneficial or harmless? Do anyone knows any microbe which is harmless or beneficial to the human body? Yeah. So basically, most microbes are not harmful, and they're actually present in water, in soil. We basically they are all present on our palms or on our faces, all over the body, and some already in our gut. But relatively harmless or more often beneficial, these microbes are always harmless and more often beneficial, and they often form biofilms. Basically, they form biofilms on our skins, biofilms in our palm. They can be seen everywhere. But with that being said, most of these microbe or the microbiome serve different functions. These include many types of microbes that we know, and they are located on various parts of our body and differ in composition. Their alteration can impact or promote disease because in their nature they are harmless, but alteration will result in diseases. So a pathogen or parasite is basically a microbe that causes disease or damage to us. How many of us have gotten several like food poison, bacterial disease, or microbial disease? The most common one is food poison. You've gone in, doctor is like, you had food poison. caused from something. Yeah, that's caused from a microbe, which is actually causing damage to you, the host, because you end up eating that microbe basically. There are actually over 2,000 different microbes that causes diseases. And there are new emerging pathogens that we've already seen. Hopefully, all of us were still, I mean, we are all alive and we experienced the SARS-CoV-19 viruses. There are also viruses which are known, such as the Zika virus. We also have the Langia virus. And then there are still new re-emerging pathogens that we've seen. Basically, one of the most common one is actually measles and polio, which is seen in the younger humans. When you are young, you don't get vaccinated, you easily contract measles and polio. And we also have diseases that once we once thought they were non-infectious. And some example is actually gastric ulcers and some gastric cancers, which are caused by Helicobacter helicobacter, which is basically a bacteria which is responsible for several ulcers in humans. And there's actually have been a link between certain cancers, bacteria, and viruses. For an example, liver cancer have been already established, which is caused by hepatitis B C virus, and we have cervical cancers, which is caused by human papilloma virus, and we have colorectal cancer, which is caused by E. coli B2 group. So now let's talk more of history. So before microscope, people believe in spontaneous generation or disease that are caused by something that we don't know is mystery. It just happened by nature, or something weird is happening we don't know, by the gods or by people. It's caused by God because we've sinned against God and all those misbelief that people had, believe that people had. So people started realizing a pattern, basically that you eat certain food, it spoils, you become inedible, and then it causes illness. And then one of the most common one was the Black Plague or smallpox, which was caused by some kind of transmission matter that people usually didn't understand. This results in a lot of speculation. People are speculating, people are switching it with beliefs and or anything they could think of. And then the first record of these was seen the Big Plague in the 6th century, which was then followed by another one in the 1800s. And then first came to us in the US around 1906. We also experienced the Black Death, which was named for darkened color of skin in systematic plague. So people really didn't know what was going on. All they know was um, diseases are happening. We don't know what is happening, and we are associating with anything we can ever think of. Followed by that, then came the breakthrough or the introduction, basically, of microbiology. So the scientist Robert Hooke was the first to observe microbes in the 1660s. Basically, he observed mold on sheepskin book cover, as we can see from here. So, back in the day, they had book covers made from sheepskin. They have molds grow on it, and he could actually see them. And then he just saw basically what appeared to be structures that were alive, but he couldn't talk much about it. But it was just it was just the era of the conspiracy. Following by that, Antoine van Leeuwenhoek also made a crucial contribution. Basically, he made a crude microscope to examine thread of fabrics in the late 1600s. And then basically he made drawings of what he could see, and he called them animalcules in rainwater, and then just scrub them from his teeth. So basically he could just see it, but he didn't know much about it. Following by then, Leeuwenhoek basically sent it to the Royal Society of London to help magnify it so that he could actually see what he was talking about. And then, followed by that, they came the discovery of pathogens and germ theory. So now people were thinking there's some organism or there's something which is little we can see unless we magnify them. And this was actually shown by Leeuwenhoek. Followed by that, Louis Pasteur basically resulted in experimenting and then disproving spontaneous generation. Basically, people thinking something is being caused by something that we don't know. He invented pasteurization and showed that human diseases could arise from infection, and disease contribute to And then his studies contributed basically to the germ theory of disease. So now Pasteur have been able to show there's something wrong, something is making us sick. Following by then, Robert Koch, also in the 1800s, discovered causative agent for anthrax. And then from there, from there was establishment of series of proof that verify the germ theories of disease. Basically, it was just seen as the Koch's postulate. Followed by that, now we have understand that there's these small organisms giving us diseases, but how do we get rid of them? How do we protect ourselves from them? We still don't know. They came the idea of sterilization. So basically, sterile, which means to completely free of all life forms, including endospore and virus particles. So just as an existence of microbes was underway, the idea of microbes could cause decay. People began to suspect that microbes could cause diseases. So John Tyndall, he found that microbes are present in dust and actually in the air, and those microbes who are actually present in the air or dust had heat resistance. That's why he discovered around the 1800s. And then Ferdinand Kuhn, he discovered and described bacterial endospores, basically bacterial present in the air, what basically John Tyndall had discovered earlier on. And then they came the idea like there should be development of methods and improved technology, basically leading to the improvement of medical microbiology and the concept of sterilization. So now we know that they are not only found in food as people were postulating back in the day, but they actually also present in air or in dust. And then there is a need to protect ourselves against them. Following that, Joseph Lister, he was the first to utilize hand washing and menstrual operating room with aseptic chemical. Back then, it was phenol in the 1800s. So basically, the technique became foundation of modern microbial control in the use of today's. Basically, he discovered you can now use chemicals such as phenol to protect or sterilize or kill microbes, basically protecting ourselves from them. Following there, people then took the concept of civilization, and this picture that you are seeing is around 1898, where you can see this nurse have a bowl, and then basically they are sterilizing everything that they are using on this patient. And then fast forward the 1910s and the 1920s, now you can see the concept of civilization have increased. As in this figure, you can see there's more bowls or more stuff you can see present here using stabilizing. And now people are actually using more like white garments and also putting on lab coats or like protective covers to protect both the patient and the ones operating on them. And here you can see similar trend. As you can see from here, people have these protective covers protecting both the patients and the people performing surgery. And then we can see that there's more improvement somewhere in the 1950s. We can see that we actually have more stabilized or a better form of surgeries being performed. We have the lamps and surgery lamps, and we have people putting on masks, and then they are sterilizing all of the instruments they are using performing surgeries. And then the 1970s, we can see things are improving better now. People have gloves, people have more protective and modernized way of protecting themselves and patients. And here in the present 2000s, you can see we've employed computers helping us perform more surgeries, helping us protect ourselves. And we actually see that civilization have now moved to the modern era. So this will be the end of class. I'm currently still working on the model page. I'll update all your assignments, I'll update everything, and I'll send you emails as the course go by for you to know when the assignments are uploaded and when you have to do it in the deadlines. If you have any questions, you can contact the TAs or you can contact me directly. Thank you and have a nice day.